Welcome to video number 4. This is Marivan, and I'll be teaching you about proteins in the body. First, let's learn about amino acids. Amino acids are the individual building blocks that are linked together by peptide bonds to create a long peptide chain that ultimately builds proteins. So, how many amino acids are there? There are 20 different amino acids. As shown by the table, each amino acid can be identified with either a 3-letter code or 1-letter code abbreviations. Each amino acid has the same basic structure of having an amino group or N-terminus and a carboxyl group or C-terminus on either side of an alpha carbon and hydrogen as its backbone, but differing only in the R group or side chain as indicated by the letter R. For instance, the amino acid alanine has an amino and a carboxyl group, but has a specific side chain of a methyl group that is just an alanine. Therefore, proteins are a construction of a long chain of amino acids that perform a variety of important duties in our body. To name a few, one example is hemoglobin, which transports oxygen and carbon dioxide in the body. Another example is an antibody like immunoglobulin, which protects the body from bacteria or viruses. Lastly, insulin has a hormonal function of regulating glucose in the body. There are 4 levels of protein structure. The first level is the primary structure. It is simply the sequence of amino acids joined together by peptide bonds. The second level is the secondary structure. This is the shape that the protein chain starts to fold into when there is an interaction of hydrogen bonds between the C and N terminus. As a result, the protein can fold into 2 shapes, a coiled shape known as an alpha helix, or it can look like pleated folds called beta sheets. The third is the tertiary structure. It is the 3-dimensional shape that occurs when the secondary structure folds within one peptide chain due to side chain interactions within itself. The fourth is the quaternary structure. This arises when multiple polypeptide chains fold together because of side chain interactions between the different chains. Now, let's explore a specific protein that is found in the body, glyceraldehyde 3-phosphate dehydrogenase isoform 1, or abbreviated GAPDH. As indicated by the suffix ase in the word dehydrogenase, this tells that this protein is an enzyme. Remember, the enzymes are made of proteins and are biological catalysts that help our body process important reactions by speeding up chemical reactions in the body. Enzymes are fabricated very uniquely with a lock and key construction to perform a specific duty. As shown here, the substrate acts like the key that fits perfectly into the active site of the enzyme or like the keyhole in a lock. Here is a microscopic view of the structure of this enzyme. Notice that this protein has alpha helices and beta sheets as its secondary structures. There are 4 different peptide chains present as denoted by the different colors in the diagram on the left. Because it has multiple chains folded together, it has quaternary structure. Within this protein are some NAD+ molecules. NAD is a coenzyme that participates in reactions. NAD+ is the oxidized form of NAD and is an electron carrier or energy storage molecule in the low energy state. This protein also contains the inorganic phosphate molecule, Pi, which plays an important role in the formation of ATP, which is the ultimate energy carrying molecule in our body. This protein makes up about 2 million molecules per cell and has a main function involving the metabolic processes in the catalyzation of glucose during glycolysis. Recall that glycolysis is a 10-step process where glucose is broken down at the expense of 2 ATP and the absence of oxygen to get 2 NADH, 2 ATP, and 2 pyruvate. Specifically, this enzyme does its magic during the last half or start of the payoff phase at the 6th or oxidation step of glycolysis. Here, NAD+ is reduced to NADH, the higher energy form. Glycolysis occurs in the cytosol or intracellular fluid found inside eukaryotic cells. This enzyme also performs other non-metabolic functions such as the activation of DNA transcription or apoptosis, where the cell literally commits suicide, in vesicle transportation from the ER or endoplasmic reticulum to the Golgi apparatus, and speeding up exonotransport. Because of its multiple functions, this protein moves between the cytosol and the nucleus of the cell. RNA and protein synthesis occurs in two steps, DNA transcription and RNA translation. But in this case, we'll be working backwards as we start with the list of amino acid sequence for this protein and work our way to get the DNA code. We will use this table to reveal the one-letter code for the first 10 amino acids in the sequence. This table shows the three-letter and single-letter codes. Here, the first amino acid in this sequence has the letter M. The letter M on the table refers to methionine, so I will write the three-letter code of MET to represent this amino acid and a dash to separate each amino acid. The rest of the amino acids will follow the same pattern. G for glycine, K for lysine, V for valine, another K for lysine, another V for valine, G for glycine, again V for valine, N is for asparagine, lastly G for glycine. RNA translation occurs in the cytosol or cytoplasm of the cell. It is a process that decodes the mRNA into amino acids to make proteins. But remember that we are working backwards, so the arrow will be making its way up to DNA. Now let's translate these 10 amino acids we just previously attained into RNA code by using this table. We'll be looking at the amino acid columns to look for the amino acid code in our list. The first amino acid is MET. As I look onto the left column, there is only one code that says AUG, so this is the code I'll be recording down. Now it's just repeating the same process for each amino acid. The next amino acid is GLY. On the left column are 4 possible codes, so I'll use GGU. LYS has 2 codes, so I'll go with AAA. For VAL, GUG. For LYS, AAG. For VAL, GUA. For GLY, GGC. For VAL, GUU. For ASN, AAC. And for GLY, GGG. DNA transcription occurs in the nucleus of the cell. This is a process where DNA is copied into mRNA. But in this case, we are continuously working in reverse mode from transcribing the mRNA into DNA code. The bases in RNA are adenine with uracil and guanine with cytosine. In DNA, adenine is always with thymine and guanine with cytosine. Remember that for base pairing, DNA only differs from RNA by one nucleotide base. So, to transcribe the RNA into DNA code, every U in the RNA code will get swapped with the letter T to get the final DNA code. That's it for today. Thank you for watching.